Let's start with the ever-time favorite in bioinformatics, differential gene expression analysis. So what is differential gene expression analysis? Before we get into this, let's talk about what is DNA, what are genes, and what are we studying. So DNA, as you all know, is a genetic makeup, and DNA comprises of sections which code for genes. And these genes code for different products. For example, a gene codes for a protein, it's called a protein coding gene. And if a gene codes for a tRNA, it's a tRNA coding gene. If a gene codes for rRNA, it's a rRNA coding gene. Now, what are we doing in differential gene expression analysis right now? We are trying to identify the genes which are differentially expressed, which means which are differently expressed in different tissues or different conditions or different scenarios. Now, how do we study this? We try to identify two sets of people or two scenarios. The first scenario would be healthy individuals. The second scenario would be affected individuals. So, from the healthy individuals, we need to get the samples, and from the affected individuals also we get the samples. Now, which samples we are talking about? Either we can take the blood, in some cases, if possible, we can take the tissues. But most easily available sample is the blood. So what do we do? So, from the healthy individuals, assume we have 10 healthy individuals. From these 10 healthy individuals, we collect the blood samples. And now, what are the cases? For example, let's consider a scenario about people who are affected with cancer. Same kind of a cancer, same stage of cancer. Now, we have taken 10 blood samples from these 10 cases as well. From these blood samples, because we are interested in differential gene expression analysis, we collect and isolate the RNA. From this RNA is a complex of mRNA, rRNA, tRNA, siRNA, all types of RNAs. From this, we extract only the mRNAs by using poly A tail bites, because mRNA generally ends with a poly A tail. Why are we isolating only the mRNA and not others? Because mRNAs are the ones which code for the proteins. So, we are trying to estimate how many copies of the mRNA are present in the normal individuals when compared to the healthy individuals, which indirectly tells us how much of protein of that mRNA is produced in this set of individuals. So, this is what we are going to do. Now, because we are into bioinformatics, we are not going to do the isolations, we are not going to do the testing of this quality of the RNA isolated. Once the RNA is isolated, it goes into a sequencer, and from the sequencer we are going to get a set of files. These set of files are called the FastQ files, which are analyzed in-house, and using this information from the FastQ files, we try to predict which gene is expressed how much in each of these individuals, and then we come up with a comparative case study. Now, let's get into the document which we have made for further details. So, what is differential gene expression analysis? Trying to identify the expression of genes whose expression is different between two or more conditions. What are those conditions? Condition 1, healthy tissue versus diseased tissue. Condition 1, treated tissue, drug treated tissue versus a control, and a wild type versus a mutant. So, what is the ultimate goal? Our goal is to identify genes upregulated in one condition and downregulated in another condition. So, what is the actual overflow looks like? We start with the FastQ files. We check the quality of the FastQ files. We trim the reads if the quality of the sequences is bad. Again, we recheck the quality to make sure that the bad quality reads are removed. Then we align these reads to the genome. There are different alignment tools. In this current example, we are going with Bowtie. And then we have another tool called SAM tools, which convert to SAM to BAM. And then there are some intermediate steps followed by transcript assembly and quantification. This is the step which tells us which transcript is how much in each of these tissues. And from this quantification, we generate a count matrix. This is the heart of the experiment. Once you get the count matrix, what are we going to do? We use DSEC2 and R package in order to perform differential gene expression analysis and followed by visualizations and other stuff. Right? Now, let's get into the next part. First, let me show you how does a FastQ file looks like. This is a PuTTY terminal which has two files in this particular folder. The SRR ID indicates the ID for every sample which is deposited in a resource from where you can download, an ID would be given. That's called an SRR ID if the data is downloaded from NCBI SRA database. How does a FastQ file looks? FastQ means a faster file with the quality scores. What is _1 and _2? The same sample file have same naming, only 1 and 2 are different. What does that mean? _1, _2 means the sequencing data is from a paired end sequencing. You have two sets of reads reading the same bit of a DNA sequence. So that's called paired end. Single end is also there, but currently we are talking about a paired end sequencing. Now, in this particular file, every sequence is represented in terms of 4 lines. The first line is the name of the sequence and the length of that sequence, followed by the actual sequence which is produced from a sequencing machine, and then a plus line which indicates that after this line you have a quality line which determines the quality of that base. For example, this is the quality sequence. How do you know whether it's of a good quality or a bad quality? Every base, when it is called, the probability of that being A. For example, the probability of that being A, they have a quality assigned. This is a set of ASCII characters which ranges from 0 to 40. Now, to get more information about it, let me just show you a quick help. FastQ Wikipedia, which gives the information. And this is the quality score. The quality score, these are ASCII values, are given from an exclamatory mark to a tile. The tile being the highest quality and the exclamatory mark being the lowest quality. So most of the sequences, when we get, each of the base are represented in either in terms of letters or in a tile. Okay, if for example, if you see this particular thing, everything is of a good quality. So the quality of the base, what does the quality mean? For example, if I give a 100 rupee note to any one of you and I ask you, what is the probability that it's a 100 rupee note? You see it, you check it and you tell that I'm 100% sure that it's a 100 rupee note. But once I blindfold you and ask you the same thing, will you be able to tell with the same confidence? No. You say that I am 80% sure that it's a 100 rupee note. In the same way, for every letter which has been sequenced, what is the probability of that letter being A? What is the probability of that letter being T? What is the probability of that letter being G? Is given, and that is the quality score, right? And the tools use this quality score to check how good or how bad your data is. To check the quality, we use the first tool called the FastQC. So these are a few steps how you download and install. When we say download and install, it's nothing big. We have to download the tool using the wget. If it's a Linux server or a Linux machine, we just unzip it. The FastQ tool, download and unzip, it works. Now, how do we execute it? So let me show you how do we execute it. First, we need to access the tool. The FastQC tool is present in Home BIB software FastQC, and this is the tool name. And now we are passing both the FastQ files, and let's see what happens. So both the files, the quality has been checked, and for each of the file it generates two output files, a HTML file and a zip file. What does this HTML file and a zip file contain? Zip file is nothing but the textual representation of whatever is there in a HTML file. Let's get into the details what a HTML file contains. This is a sample HTML file which we have been checked. A FastQC gives information about all these parameters 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And now let's get into each of the details. The name of the FastQ file, the type, in which machine it has come, the total number of sequences, how much GB of the data is there, are there any sequences which are marked poor, what is the length of the sequence, and the percentage GC. This is the basic statistics. The FastQC has 3 parameters to indicate whether the quality of the data is good or bad. If ever 3 levels, a green tick is good, a orange exclamatory, you have some problem with the data, red, you definitely have a problem with the data, right? So let's check the per base sequence quality.